And we're now about to move into what is DNA structure and how does it actually function. So we finally got into this place where through observation, over millennia, we get to a monk, and a monk comes up with these laws that statistically describe the likelihood of actually seeing a phenotype. And then we come across a tool, microscopy, that allows us to take a look at the fact that whatever these inherited particles are, they distinctly have a pattern for moving around, and they distinctly have a structure. So inside chapter 14, what we're going to do is we're going to look at what were the steps from, say, 1900 till modern day sequencing techniques for us to actually look at what DNA structure is and what sort of information it holds. Much of this information you already understand. You know DNA is made up of deoxyribose sugars. You have a double alpha helical structure with a sugar phosphate backbone, and you have 4 different nucleotides and how that's all established. What you want to pay attention to is your ability to discern what the experiments were that actually helped people figure out the mechanisms for, 1, creating DNA structure, and 2, having it function at distinct periods in time in a cell's responsibility. It starts with a story, okay? Working inside a hospital clinic, pulling the nuclei of leukocytes from pus inside hospital bandages, what Friedrich did was isolate that nuclear material, and because it was phosphate rich, okay, and acidic, he was able to give it a specific name. He called it nuclein, right, which eventually would become called our deoxyribonucleic acid, okay, that sugar phosphate backbone. The acidic nature, using the characteristics of the material, ultimately this helps us figure out how it actually functions. It would take some time before a transformation principle would actually come out of this. And this is a really cool experiment. It's deadly. It's deadly for the mice that it was practiced on. But if you get Streptococcus pneumonia, chances are it doesn't work out for you all that well either. So the question was, Frederick Griffith was trying to figure out which population of Streptococcus actually killed the mouse. Okay? And we have to have some controls. We actually have to have something that will tell us ahead of time what's going to happen. Now, a mouse can be injected with a known strain, okay, that should kill it. Now, that you might think of that as an experimental, but what you do, of course, is you put it in to the mouse with a strain that hasn't been heat treated versus a strain that has been heat treated versus an untreated mouse. So you see that even though they show you only 1 mouse here, there have to be at least 3 mice inside this experiment to draw this one conclusion. So he takes this S strain, okay, which is smooth colonies and is pathogenic. Okay, so you actually have smooth colonies of Streptococcus pneumoniae in the bacteria. Heat treat it, boil the snot out of it is what he did, right? And he stuck it inside the mouse. And you know what happened here. We changed the 3-dimensional shape of the proteins on the smooth rounded bacteria, and the mouse is just like, yeah, I'm good to go. Now, you can also throw in the fact that the R strain has rough colonies and it's not pathogenic. So we could exchange now or increase our number of mice to 4, repeat the entire experiment and demonstrate R strain didn't kill, S strain did kill if it wasn't heat treated, S strain didn't kill if it was heat treated versus our untreated mouse. And it's important you remember there's an untreated mouse in all of this because that is your control. Finally, the real experiment, okay? So when Griffith injected a mouse with a heat-killed strain and a live R strain, the mouse died. And this is when you go heat-killed plus R, and you're like, WTF? What the hell happened here? What he had done is he had discovered that some material from the S strain would ultimately be conferred over to that R strain. And in the process, it transformed the R strain into the S strain. And you've already seen this idea. I've already explained to you that you can have small pieces of genetic information that can pass between bacteria using their pili, and we call that material a plasmid. Okay, and that's what's going to actually happen here. So when we think about, let's go back to this, what Oswald and Avery would, so Avery and McLeod and McCarty would show is that the principle ultimately would have to be some form of a nucleic acid. Now, I've told you that it's a plasmid, and unfortunately that skipped 50 years worth of research when I told you that, right? But what they did is they systematically used enzymes to degrade the macromolecules, and they sequentially mixed these extracts with R strains, and they then injected this inside the mice. So they came up with a different way to ask a similar question, but by using, instead of just heat, they figured out which enzymes would break down which macromolecules. Remember, there's only 4 here: nucleic acids, proteins, carbohydrates, and lipids. And when they injected that into the mice, that's when they discover that you can now build on, you can now build on what's likely going to be the thing that, if you broke down the nucleic acids, it meant that nothing from the S strain could kill the mice when transferred into the R strain. Now we come to a little bit more of an elegant experiment where, using prokaryotes, using bacteria, I don't know if you know this or not, can be infected. They can be infected by viruses, and that virus is called a bacteriophage. Okay? And that bacteriophage literally injects their genes into bacteria to reproduce. So people wanted to figure out what that injected material was. And this is where your knowledge of chemistry and physics actually becomes really important. Bacteria were infected with a phage that was radio-labeled, okay? Where S35, remember when proteins fold and they actually take that three-dimensional shape, hydrogen bonding can hold them in place, but so can disulfide bridges. So disulfide bridges use sulfur, and what happens is those proteins are now S35 late labeled. DNA, by this point in time, it's been figured out, of course, it's a phosphoric acid, okay? So P32 can be used to build the DNA inside of those bacteriophages. And what happens is you then fed your bacteria to your bacteriophages, okay? And then you put it inside a blender. It's a really cool experiment. Right? And giving them only just enough time to see what would happen, who got into the bacteria. So the blender can't be coarse enough to mess up everything. It just has to take the phage coats off, right? And when they do that, and they centrifuge down the material, they see that only P32 has actually entered the cells, the bacteria themselves, right? Suggesting that the material that was conferring this information over here, our transforming principle with the Frederick Griffith experiment, okay, was actually a DNA molecule. So it takes 15 years here to actually get to this place where a clean experiment demonstrates that it's DNA and not proteins or not lipids or not carbohydrates that's affecting the behavior of cells. And here's the experiment for you one more time, where it's drawn out for you. Here's your S35 labeled protein coat. Here's your P32 labeled DNA. And in some instances, you could design the experiment so it's both of them together. Okay, it just depends on your skill as in this case, a bacteriologist. And what's going to happen, the bacteria are infected, you separated them out, and then you figured out where the radio label was. Okay? So it was only those with the P32 up here that actually ended up inside of the plasmid that actually demonstrated that you could transform the cells. It would take another 6 years now to figure out what is the ratio of information. You need to get to the Chargaff's rules, where the percentage of adenine and thymine and the percentage of cytosine and guanine have to be roughly equal to one another. Because what's going on, of course, is now you actually have 4 nucleotide building blocks, adenine, thymine, cytosine, and guanine, and you need to figure out the ratio. So A's equal T's, T's equal A's, and this is why today we say A double bonds to thymine, thymine double bonds to adenine, cytosine triple bonds to guanine, guanine triple bonds to cytosine. This is not that experiment that demonstrates that, but because these percentages were figured out, generally speaking, you could then figure out how to build now a state of stable DNA. Sorry about that. Let's see if I can edit that out. Together, you break them apart into two families. You have the pyrimidines, which are our uracils for RNA, thymine, and cytosine. Our purines, which are adenine and guanine. And remember, these are going to form complementary base pairs. Adenine binds to thymine, guanine binds to cytosine. But when we actually think about how they're constructed, what you see here is, here's the nucleoside monophosphate, nucleoside diphosphate, nucleoside triphosphate, and this is actually how you build, say, ATP. This is adenine here, right? ATP. Got it? Now, who cares about that? If you have this complexity, if you have this diversity in complexity, it means now you can store something inside of it. And this is how we begin to understand the structure of DNA. We take those rules and Rosalind Franklin's X-ray crystallographic work for Watson and Crick to sit down in 1953 now. This is 3 years later, okay? To say, look, we figured out that this actually has to be a two-sided structure that's holding together pieces of information. And that's what this X-ray crystallograph actually shows you. As its name suggests, a crystal has to be formed. You pass X-rays through it. Those that are the densest materials stop the film from being exposed. Those that allow X-rays to pass through expose the film, similar to how your own X-rays work when you break a bone. Now, what we end up seeing, of course, is you have this sugar phosphate double backbone here with the nucleotides that are set inside the middle. And here's that double bond I was telling you about a moment ago. And here's that triple bond I was telling you about. And what's important to see here is those two polymer strands are held together by hydrogen bonds of opposing nitrogenous bases. So pyrimidines and purines. Sort of think of it as a complementary base pairing system where the electrostatic charges here are what's actually storing the order of information. And that's, a way to introduce what you could see here. You can go take a look at this. This is the way we actually sequence DNA at this point in time using the Sanger method. This is old school stuff, doing this when I was an undergrad. You go nucleotide by using a series of enzymatic reactions. And it's important for you to see how it's done. How we do it today, though, in labs that don't have a lot of money, you would probably still continue to use Sanger method sequencing, okay? But these days, you use high throughput sequencing, systems that can sequence literally. It took, 10 years to sequence the human genome once. Now you can actually get the entire human genome sequenced with at least one pass of reading done in probably less than 2 weeks. Okay? What do you get? Well, this is a great place to pause because if this is how we actually sequence information, old school style, what you could do is then ask yourself, how does it all replicate? Okay? How does it know what to make and how to make it? I know we take for granted that it does replicate, but I want to pause here and we'll switch over then to the next topic of replication.